Hello, and welcome back to Biology with Miss Ellis. I want to kind of explain some specific examples from the notes in a little bit more detail. And so, we're going to talk about some of the different checkpoints that occur in the cell cycle. So, you're going to be adding this information to your diagrams at the end of the doc that you started yesterday. In the G1 checkpoint, this is also known as the restriction checkpoint. And basically, the cell, and I'm using hyperbole here, the cell doesn't literally ask itself questions, but this is kind of what it's going to say. Is the cell large enough? Are there enough nutrients? Is the DNA undamaged? And so if we can answer yes to these three questions, then the cell receives what we call the go-ahead signal, and the cell cycle continues. So it's going to proceed now to copy the DNA, move into G2 and mitosis as long as those other checkpoints are met. If we answer no to any of the questions, then the cell does not receive the go-ahead signal and it'll revert into the G0, which is a non-dividing state. So let's talk a little bit about MPF. MPF stands for the mitosis promoting factor, and this serves as the signal for the cell to actually begin division of the nucleus and all the things that follow during mitosis. So you can see that MPF fluctuates. It starts to increase, and when it's at its peak, that's when we begin mitosis. And so you can see that through G1 during interphase is when MPF is at its lowest. Now, take a look at cyclin here for a second. Notice that cyclin never actually goes away, but it also does cycle. That's because cyclin is always present in cells, and it gets regenerated, reused, which I'm going to talk a little bit about more in just a second. But as we proceed over time, and you can see here that there were 1, 2, 3 cell cycles in this particular example. As the MPF increases, the cell enters mitosis, and MPF is the go-ahead signal for nuclear division. And again, I'm going to explain MPF a little bit better in the next little section. So at the height of MPF concentration, that tells the cell, okay, let's do this. Let's do all the things that happen during pro, meta, ana, and telophase. We're ready to separate the nucleus. So let's talk about this process in a little bit more detail. So you have this diagram, and I've kind of created a little chart where you can take some notes. So let's start here at number 1. So number 1, we see that the concentration of cyclin is accumulating. So the concentration is getting larger. So as cyclin increases, the concentration of cyclin increases during late S, so after we copy the DNA, and it continues through G2. So during that last spurt of growth before we actually start mitosis. So the cell's building up these cyclins. Now in 2, we've also got cyclin dependent kinases. So we've got CDKs and cyclin. These two things combine to form this MPF, the mitosis promoting factor. It's just a large protein complex. And so once the CDK and cyclin combine to make this mitosis promoting factor complex, once we've made enough of MPF, we initiate mitosis. Now, MPF, once we have it, it's going to phosphorylate a lot of different proteins in a cell. And of course, phosphorylation is just a fancy word that means we're going to add a phosphate group to a whole bunch of other molecules. This causes a signal cascade, and then that signal cascade is going to cause all the responses for mitosis. So formation of spindle fibers, breakdown of the nuclear membrane, aligning of the chromosomes, spindle fibers attaching to the chromatids, etc., etc. Now, once we have reached mitosis, about anaphase or so, the cyclin will begin to degrade, and that happens during anaphase, which basically begins to bring an end to the mitosis portion of the cell cycle. And so the new cells that are formed are in G1. So they're brand new cells. They're going to be in G1, hanging out, doing their thing, basically just being an interphase. Now, the CDK, if you think back, or you can even look back at the previous diagram, You know how CDK like never went below the graph? That means that the concentration of CDK never reaches zero, it never totally goes away. That's because CDK is recycled back into the cycle and it begins to accumulate again. So it's the same thing. We're building up, building up, building up until we get enough. It combines with cyclin. We have the mitosis promoting factor, and this whole thing starts over. Okay, I'm going to take a breath for a second. So, normal cells have a couple of different properties that we've kind of talked about. One of those is anchorage dependence. What that means is cells require a surface for which they can divide on. So they have to either be attached, like in a lab, they have to be attached to a culture jar or a petri dish. And like in this instance, they have to be attached to something. In real life, they can be attached to other cells, which we would call tissues. So in normal cells, they also exhibit this property called density dependent inhibition. So you'll see in the petri dish or culture jar, call it what you will, you got a nice single layer. And then if we remove or scrape some of those cells away, they'll divide to fill in that hole, but they never divide any more beyond that single layer. So inhibition stops the cells from reproducing and making too many. So you've got your single layer. If it gets scraped away, the cells are replaced. But it never goes beyond that single layer because cells can sense if there may not be enough nutrients. There's no reason to continue to divide. This could kind of represent an injury, if you will. We replace those cells, everything's good to go. Cancer cells, however, divide beyond that single layer. They keep dividing. They don't care. It's too crowded, don't care. We're going to keep dividing and keep dividing and keep dividing. So they divide beyond that single layer and they form clumps of overlapping cells. So cancer cells do not exhibit anchorage dependence. They do not exhibit density inhibition. They're just going to keep dividing no matter what. And then finally, let's just kind of take a look at normal cells versus cancerous cells. So in this first diagram, you can see that the cells are fairly, you know, basically the same size. They're kind of, spread out or whatever. Cancer cells will continue to divide and grow no matter what. These are nicely shaped. They're spread out. They exhibit density dependent inhibition. These do not. Here you can see that the normal cells are all relatively the same size and shape. Cancer cells show variation in shapes and sizes. You can see here that this normal cell, the nucleus is fairly normal. Here in the cancer cell, it's larger and darker than normal. Here in this normal cell, the chromosomes are nice and even. In this cancer cell, there's an abnormal number of chromosomes and they're arranged in a disorganized fashion. And again, here, the normal cells they have boundaries. They're, not dividing beyond their specific area. Cancer cells are clusters of cells with no boundaries. So I hope you found this helpful. And I just wanted to kind of go over some visuals that you might see on the AP exam with regards to controlling the cell cycle. Hope that